Hi, everyone, and welcome to track one of our virtual symposium on neuroimaging indicators of brain structure and function. This track will focus on opportunities and obstacles on the road towards reaching our goal of precision multimodal structural magnetic resonance imaging, or MRI. We'll have 4 talks that each describe machine learning approaches to solve an important scientific problem related to precision MRI. These approaches have multiple interesting benefits, from enabling neuroscientific study of clinically acquired images to better understand the human brain, to increasing knowledge about the human brain over the lifespan, to enabling quantitative brain measurements in the clinic. These precision MRI approaches have the potential to help us understand factors affecting brain health, detect brain abnormalities, understand brain aging, and more. But first, multiple challenges need to be solved. The 4 talks you are about to hear describe cutting-edge machine learning approaches to address particular challenges in precision MRI. The talks will be given by researchers at all stages of their scientific careers. Our speakers include a graduate student from MIT, as well as early and mid-career researchers based at academic medical centers. As you are watching these talks, you are welcome to enter questions using the Q&A feature in Zoom. So please enter the question whenever you think of it, and we will answer questions as time permits at the end of the talks. Thank you very much for joining us, and now I'll introduce our first speaker, Nalini Singh. Nalini is a PhD student at Harvard MIT program in Health Sciences and Technology. She works in the Medical Vision Group at MIT's Computer Science and Artificial Intelligence Laboratory, CSAIL. Her primary research interests are in medical image reconstruction and analysis, signal processing, and inverse problems. Previously, she received a bachelor's degree in electrical engineering and computer science from MIT. Thank you for joining us, Nalini. We're excited to hear your talk.
Thanks, Lauren, for that introduction. Today, I'm going to tell you all about how we can use machine learning to get really high-quality MRI reconstructions. So the whole process for getting and using an MRI scan starts when the patient comes into the scanner. And when that happens, the data that we acquire is what we call K-space data. And this is data that lives in the frequency space or the Fourier space. And there's actually an extra step which is needed to convert that K-space data into an image which a clinician or a researcher can look at and use. Typically, that step involves something like an inverse Fourier transform. Once we have the image, there's a few extra optional downstream steps we can do. So you might do some higher level analysis in order to get something like a segmentation of the brain. Or once you've done that, you might even go further and do some extra analysis to get a metric which tells you something about how healthy a particular patient or a particular population is. So this is something like analyzing the volume of a structure that you've segmented and then understanding how that changes with age. So in today's symposium, people are going to talk about how you can use machine learning at all of the different steps along this pipeline. But my talk is going to focus on one of these first steps, which is going from the K-space data to the image space data. And in particular, I'm going to tell you about how we can get really good quality images, even when the K-space data that we collect is imperfect in some way. So in the case where we start off with perfect K-space data, it's pretty straightforward to reconstruct a high quality image. All we have to do is apply an inverse Fourier transform to the data that we acquire, and then we get a nice looking image like the one on the right. The issue is if we acquire imperfect K-space data, it becomes a little bit less clear what we need to do to the data in order to get a high quality image. So what I'm going to do now is tell you about 3 reasons why we might end up with imperfect K-space data. And then later in the talk, I'm going to tell you about our proposed methods for fixing this problem. And I'm going to start by saying all of the examples I'm going to show you in this section are really extreme examples of the type of corruption you might see. And this is just to illustrate sort of qualitatively the types of artifacts that you would get as a result of imperfect K-space data. And later we'll talk a little bit more about how to fix them. So one of the first reasons you might end up with imperfect K-space data is undersampling. And this is an imaging choice that we make when we're designing sequences in order to get an image. So doing an MRI takes a pretty long time, and that's undesirable for a couple of reasons. One of which is that it's uncomfortable for the patient while they're in the scanner. Another one is that the longer time the patient is in the scanner, the more opportunities there are for the patient to move around while you're taking the image. So in order to avoid that, one choice we might make is instead of acquiring all of the data at all of these points in K-space, we might just choose to collect data at some subset of those points. So for example, you might choose to just collect data at each of these blue lines in K-space. If you do that, and then you just apply an inverse Fourier transform to the data that you've collected, you'll end up with an image which looks like the one on the right over here. And you can see that you lose pretty much all of the important structure in the image that you were trying to reconstruct. So another reason you might end up with imperfect K-space data is motion. So let's say the patient comes in and they get in the scanner and they start off in some position like this on the left, and you acquire some subset of your K-space data while they're in that position. Then partway through the scan, the patient moves, probably unintentionally, and you get a patient that's in a position like this. So this is pretty different from what it's like in the first position, but you continue acquiring K-space data as part of the same scan. So at the end of this whole process, you end up with a set of K-space data, which is sort of this mix between the patient in one position and the patient in the other. And you can do an inverse Fourier transform on that data. But if you zoom into that result, you end up with this really horrible warbling effect. And just like in the previous example, you've lost a lot of the structure in the image that you're interested in. The third reason I'm going to tell you about that you might end up with imperfect K-space data is the case of noisy measurements. So this typically happens when you have a magnet in your scanner, which isn't quite as strong as you'd like it to be. And you end up with noisy K-space data, which looks like this. And if you do an inverse Fourier transform, that corresponds to a noisy image, which looks like the one on the right. And just as in the previous two examples, we've lost a lot of the really important structure in the image here. So in all three of these cases, even though we've started off with this corrupt or imperfect K-space data, we still want to be able to reconstruct a really high quality image. And the idea is that this is where we can really use machine learning as a complement to what we already know about the inverse Fourier transform in order to correct for some of the effects that we've dealt with due to these three reasons. And this isn't really a new idea. People have been using some techniques from machine learning for quite a while in order to do this type of reconstruction. And that's why typically the images that you get from the scanner today don't look nearly as bad as the images that I just showed you. Many of these previous approaches are based on iterative optimization approaches. So at each stage of this approach, you estimate a successively better guess of what the reconstructed image should look like. And these approaches work reasonably well, but they're very time consuming. So these approaches might take several thousand optimization steps, and each step itself might be somewhat slow. So overall, this whole process can take several hours to even a day or two in order to reconstruct a single image. So as a result of that, there's been a lot of recent interest in using neural networks instead to do this problem. And with a well-trained neural network, you can get a high quality reconstruction over the course of just about a few hundreds of milliseconds. So most of the approaches which have been developed for using neural networks to solve this problem operate in the image space. And what I mean by that is that typically these networks are comprised of layers which perform convolutions, which are these small local operations on patches of images or on features derived from those images. And these approaches work all right, and they're very fast, but they're a little bit unsatisfying in the sense that they don't directly capitalize on relationships which we know exist in K-space. And to motivate this a little bit more, I'm going to ask you to think about just a single example of an image, which I'm showing up on the screen, and its corresponding K-space data. And now what I'm going to show you is what happens if we corrupt just a small patch of data within the K-space. So if we just flip out that small patch of K-space, there was a slight shift in the image. And I'll show you also the difference between this image after the corruption and what the image was before. So you can see that all of the errors that we've introduced in the K-space are pretty well localized here in the K-space data. But once we move over to the image space, the results of these errors are spread out and dispersed throughout the entire image. So there's some intuition here that it might be a bit easier to work in the K-space to fix these issues, because we would just need to fix these few pixels in the K-space. Whereas if we wait until we've gotten to the image space, we need to really do a lot of fixing across the entire image. So with that motivating example in mind, we really wanted to answer this question, which is, if you do want to use a neural network in order to do high quality MRI reconstruction, what should be the core operations which comprise the layers which make up that neural network? And to give you a little bit more intuition towards the solution that we're going to propose for this problem, I'm going to show you some data about the correlation structure that's present in both the K-space and the image space for a particular large data set of brain MRIs. So here's a visualization of that correlation structure. And on the right is an explanation of what exactly this visualization means. So there's a circled pixel, and that's a pixel of interest. What we want to understand is, how do other pixels in the image relate to this circled pixel? The rest of the pixels in the image are colored either red, white, or blue. And a red pixel indicates that across this large data set that we're studying, there's generally a positive correlation between pixels at that location and pixels at the circled location. Similarly, a white pixel indicates that there's zero correlation across that data set between that pixel and the pixel that we're studying. And a blue pixel indicates a strong negative correlation between that pixel and the pixel that we're studying. So now if we take a look at this visualization on the left, you can see that for this particular pixel of interest, a lot of the subcortical regions are highlighted as being pretty closely correlated with the pixel that we're interested in studying. Now, if I animate this visualization and move around that pixel of interest, so you get a sense of how these correlation patterns vary across the entire image, it's pretty clear that at different locations in the image, it's not always true that the subcortical regions are the ones which are highlighted. But one thing that is true across all the locations in the image is that there's at least a small local patch where there's a pretty strong correlation between the surrounding pixels and the pixel of interest. And so this gives us a sense that the image space convolutions I was talking about earlier are actually a pretty good approach for getting some information about the pixels that we're trying to estimate. Next, I'm going to show you the similar visualization, but I'm going to show it to you for the K-space data of these brain images instead of the image space data. And I'll give you just a second to look at it. And one of the first things you probably notice here is that very different to the images on the left, we have sort of these two different patches which seem to be circling around. That corresponds to this conjugate symmetry characteristic, which is generally true of real valued K-space data. And I'm actually going to ask you not to focus on that for this example. Instead, what I'm going to ask you to look at is within this individual circle of interest, which is moving around your screen, you notice that just like in the image space case, there's a pretty localized patch of strong positive and negative correlations with the pixel of interest. So in addition to the image space convolutions that we previously saw might be warranted, what this tells us is that it actually might be really useful to do local convolution-like operations in the frequency space, which capitalize on these local K-space relationships. So with that in mind, we set out to design some core layer structures for a neural network, which incorporate both convolutions in the frequency space and the image space. And here I'm showing you one example of those layer structures. So actually this entire diagram represents a neural network, but I'm going to ask you to focus on this section, which is in the light purple box. And I'll refer to this as the alternating version of the layer. So within this diagram, the red squares represent features which live in the frequency space, and the blue squares represent features which live in the image space. And what happens within an individual layer is that you start off with a frequency space feature, you apply some sort of convolution in frequency space, and then you move over to the image space via an inverse Fourier transform. Once you're there, you have some image space features, you apply a convolution in the image space, and then you go back to the Fourier space. And you keep alternating between these two spaces as you move down the network until ultimately you end up with some high quality image reconstruction. We also designed another version of a layer which incorporates both frequency and image space convolutions. And in this case, the key difference is that really both of the types of features are processed in parallel. So again, you have some sort of frequency space features, but you also start off with a set of image space features in this case. And then as an intermediate in this layer, you learn some linear combination of versions of these two sets of input features. So for example, up here in this frequency space branch of this layer, you start off with the frequency space features, and you learn some linear combination of those with the Fourier transform of the image space features that you started with. You do a similar version of all of this in the image space down in this bottom set of the operations of this layer. And then for both of the intermediate values which live in the frequency space or the image space, you apply a convolution in the appropriate space. So at each layer in this network, you keep these parallel representations of both frequency space and image space features, which are mixed at each layer. We wanted to understand how both of these layers compare to sort of these baseline cases where either you just operate in the image space or you just operate in the frequency space. So what we did is we compared networks which look like this to corresponding networks which either have just this top row of operations, so those are just frequency space convolutions, or just this bottom row of operations, so those are just image space convolutions. And next, I'll show you some qualitative results from a wide set of experiments we did comparing these sets of architectures. So we did these experiments on 2D slices from T1 weighted brain MRIs from the Alzheimer's Disease Neuroimaging Initiative. And we had several thousand training slices, several thousand validation slices, and then we held out about 100 test slices, which we used to evaluate all of our results. And there was no overlap in subjects between any of these sets. And we performed 3 sets of simulation experiments on this data set. So the first was that we simulated undersampling with a factor of 4. So this means that we only simulated acquiring data at 25% of the points in K-space. The second experiment we did was to simulate motion corruption. And so we said, for roughly 3% of the lines within a particular K-space, we would allow translations of up to 8% of the field of view or rotations up to 15 degrees. And then finally, we simulated noise. So we added zero mean complex Gaussian noise to all of the values in K-space with a particular standard deviation. And here are some qualitative results from each of those experiments. So the leftmost image you see here shows the ground truth image, and this is the thing that we're trying to reconstruct with our networks. The thing on the right shows the input to these networks. And so this is a pretty highly corrupted version of the data. And in this case, I'm showing an example from the undersampling experiment. There's also a patch highlighted, which we'll zoom into a bit later. Next, I'm showing you the results from the two baseline networks. So this is a network which is operating just in the frequency space. This is a network which is operating just in the image space. And then finally, these last two columns show you the results from our networks, which include layers which have both of those types of convolutions in the frequency space and the image space. So now let's zoom in on that patch of interest. And the second row shows you the patch itself. The third row shows you the difference between the reconstructed patch and the ground truth patch. So brighter colors in this third row mean that you have more error. If you look pretty closely at these patches, you can see that there are all of these small detailed features which we're able to reconstruct quite well with the joint networks, which you totally lose access to with the image space or pure frequency space networks. Also, if we look at the reconstructed Fourier space data, qualitatively, you can see that this data is much more smoothly reconstructed in both of the joint networks, whereas in the pure image space network and the pure frequency space network, you still maintain a lot of the sort of streaking artifacts that you can see in the input. So here's a similar results visualization for the case of motion corruption. And if you look at this input here, you'll notice that a pretty big difference between the last example at this location and this example is that now we have this sort of stamped moved copy of the image of interest. And that's the motion effect that we're trying to correct. So again, if we look at the image patches here, you can see that there are a lot of really high frequency features, which we're able to reconstruct quite well, that the pure image and pure frequency space networks lose out on. And similarly to the previous example, if you look at the frequency space data, we're not perfect in reconstructing here, but compared to the image space K-space and the frequency space K-space, we do have a more qualitatively pleasing result, which fills the space a bit more uniformly. Finally, here's a qualitative example from the noise corruption case. Just as before, now we have the noisy data here in the input. And just as before, if we look at the image patches, there are a lot of these really high frequency features which we're able to reconstruct quite nicely, which you're not able to see in the pure frequency or pure image space case. And once again, if you look at the frequency space data, we fill the space a bit more symmetrically and nicely compared to the other two examples. So these are all just individual examples of reconstructions, but we can also look at quantitative results across the entire test set. So in the left column here, I'm showing results from the undersampling example. In the middle column, I'm showing results from the motion corruption example. And in the right column, I'm showing results from the noise corruption example. For each of these plots, the x-axis shows the index of a particular subject within the test set, and the y-axis shows the error in the reconstruction. So for all of these plots, a lower value means that we have a better reconstruction. Each dot within each of these plots represents one reconstruction from one of the networks. So the purple dots of both shades represent reconstructions from the joint networks, which include both frequency space and image space convolutions. The blue dots represent reconstructions from the networks which operate just in the image space. And the red dots represent reconstructions from the networks which operate just in the frequency space. And you can see pretty uniformly across all of the subjects, across all of these sets of experiments, the joint networks do a much better job reconstructing compared to the other two networks. So before I conclude here, I want to thank my really awesome collaborators, including Eugenio, who's actually going to give the next talk following this one, as well as my generous funding sources. And I want to leave you with two main conclusions which summarize my talk. The first is that at a high level, we can use machine learning to get really high quality image reconstructions, even when we acquire case-based data, which is severely corrupted. And the second is that if you're going to use a neural network in order to do this type of reconstruction, you really want to use a neural network where the core layers are comprised of operations which include both frequency space and image space convolutions, as opposed to using a network where the layers perform operations just in one space or the other. That concludes my presentation, and now I'm going to pass it back to Lauren, who will introduce the next speaker.
Thank you very much, Nalini, for that great talk. Now I'll introduce our second speaker, Eugenio Iglesias. Eugenio is a part-time lecturer at University College London and a part-time lecturer at the Martinos Center in Boston. He is also a research affiliate at MIT. His main research interest is the construction of computational atlases of neuroanatomy using high-resolution ex vivo imaging and their application to segmentation of brain MRI scans of any resolution and contrast. Thank you very much, Eugenio. Please go ahead.
Thank you very much for the kind introduction, Lauren. Whoops. See, there you go. So neuroimaging tools, yes, they have enabled us to learn about the human brain in health and in disease by automatically analyzing brain MRI scans from living people, both healthy controls and diseased subjects. One example of a popular analysis is image segmentation using packages like FreeSurfer or FSL. So starting from an MRI scan like this, you can automatically get it labeled, and you can use this segmentation to compute, for example, the volume of different structures and look at how this volume changes with, say, in normal aging or in Alzheimer's disease. Another popular analysis from segmentation is cortical thickness, which is a powerful biomarker, again, in many diseases. And another popular analysis is image registration, so deforming one brain scan to another. A popular tool for this is, for example, ANTS, but there are many others like Elastics or NIFT-Reg. And you can use it for example, longitudinal analysis, where you say, well, I'm going to compare two scans from the same subject at different time points, so I can deform one to the other and look at this deformation to see if things are shrinking or they are expanding. You can do this more formally with population in a very popular analysis called tensor-based morphometry. So this is an example of aging in healthy controls and in Alzheimer's disease, where red is expansion, as you see, like in the ventricles, much more so in Alzheimer's disease. And blue is contraction or atrophy. So you see a lot of atrophy in the middle temporal lobe in Alzheimer's. And registration is also important for multimodal analysis, right? Like when you want to look at two modalities simultaneously, you want to put the pixels of one modality on top of the other one. So that's a registration problem as well. So, and these tools are super useful for this and many other analyses. But the problem here is that they normally require images, MRI scans with isotropic resolution, which are the ones that we normally acquire in research, right? Sometimes they even have requirements in terms of contrast. For example, FreeSurfer runs on T1 scans, ideally acquired with an MP-rage sequence. So, research scans are isotropic. Many of you are used to seeing this. So it doesn't matter if I look at the axial, coronal, or sagittal view. It looks good in all of them. But, in the clinic, this is not the type of scans that we acquire. They're normally sparse, meaning like they have like bigger separation between slices, and they're often multimodal with different MRI contrasts acquired in different orientations. So this is an example of a sagittal T1 that looks nice in sagittal plane. And this is an axial flare that also looks great in plane. But if you look at it in the coronal view, it looks pretty terrible, right? So now the question is, can we take a bunch of scans from a clinical exam, put it in a cocktail mixer, shake it a little bit, and get a 1 millimeter MP-rage out of it? And it sounds like it should be possible, right? Especially when you have scans acquired in different orientations. Once you've done that, you can analyze it with standard tools, say FSL, FreeSurfer, SPM, whatever you want, your favorite package. And this has a lot of potential in two different ways. One is that you can go back and analyze like boatloads of scans that are currently sitting in packs around the world. You can look at these data sets retrospectively with very, very large sample sizes. And the other thing is that it represents a step towards quantitative clinical neuroimaging, whereby instead of like eyeballing qualitatively measurements of, say, I don't know, ventricular expansion or hippocampal atrophy, you can actually get volumes measured with say, FreeSurfer. To do so, we need to solve two problems. One of them is the difference in resolution. So this is a super resolution problem. You're trying to go from slices with large spacing to an isotropic scan. And the other problem, of course, is the contrast synthesis, right? You want to go from all these different, say, said flare T2, maybe turbospin echo T1 to a canonical reference contrast, typically MP-rage, which is what makes all these packages happy. So you could think, hey, I've heard that convolutional neural networks, CNNs, they can't do anything. And so why don't train a CNN? And I'm like, yeah, okay, fine. That sounds fine. You can do this. There's no problem. And so if you have the right training data, though, so if you give me, images that look like this, like the ones I showed you earlier, and you have matching acquisitions of isotropic, say, MP ranges, you could train a CNN to learn how to turn these guys into something that looks like this. And that's perfectly fine. No problem with it. But the thing is, you would need to train a separate CNN, and you would need to gather training data for every combination of MRI contrast, resolution, and orientation that you're interested in. And if you're looking back into this, like images taken from a PAX, this is an absolute nightmare, right? Because the number of combinations is huge. And sometimes getting this training data may not even be possible, or it may be super hard. Like, for example, these turbo-spin echo sequences used in the clinic, or Hyperfine, which is this portable MRI system, which I'm going to go back to later on. It may be very, very difficult to get matched 1 millimeter MP ranges. So, all right, this is our proposed solution. This is a paper that's going through a round of revisions right now in your image with a very long title, which is almost identical to the title of the talk. But the idea is training these networks with synthetic data. And the approach, we called it SynthSR, SR for super resolution. Synthetic data, the advantage is that you can simulate any contrast and resolution you want without having to acquire any new data sets. So the idea is that at every mini batch, so at every step of training a convolutional neural network, you start from a pool of 3D segmentations, which we also call label maps. And you select one at random. This is pretty standard. And it's also standard to do some geometric image augmentation. So you randomly deform this label map to increase the variability in your training data set and improve the generalization ability of the CNN that you're training. And from this deformed label map, you use a simple likelihood model, which is a Gaussian mixture, to generate this synthetic high resolution HR. It's for high resolution. Synthetic scans, fake scans. So these are like high resolution intensities. They don't have to look like real scans, but I'm making them look like a T2 and a T1 here, just for the sake of this presentation, but you wouldn't have to. And then what you can do is to take one or more of these like high resolution synthetic scans and introduce artifacts, including noise, including bias field, and crucially for this talk, for this work, you can simulate the lower resolution, right? So I'm taking this high resolution T2 and I'm making it look like a lower resolution axial T2. So it looks ugly in coronal plane, just as the images I showed you earlier, right? So you can do this at every mini batch, at every step, to generate data, and then train a CNN to do a lot of different things within this framework. So something you could do is, for example, you could learn how to segment high resolution T1s if you wanted to, like using these guys as input and the segmentation as output. You could also learn how to go from your low resolution intensities to your high resolution segmentation, which is known as partial volume segmentation. You could also do contrast synthesis, learning how to go, for example, from T2 to T1. You could do super resolution, learning how to go from low res to high res, from LR to HR. In this talk, what we're trying to do is simultaneous super resolution and synthesis. So you could learn how to go, for example, from a low res T2 to a 1 millimeter T1. And the thing is, this is what we tried in the beginning, and it kind of works, but because these synthetic images are a bit cartoonish, we actually got better results going back to the real T1s. So these segmentations from our training data were made on actual MP ranges. So you can define this reference contrast as the contrast of your training data set, and then learn how to go from your synthetic low resolution images, which can be, like I said, multiple channels, for example, a sagittal T1 and an axial T2, and learn how to get your 1 millimeter MP range. So this is how we train convolutional neural networks for joint super resolution and contrast. So I'm going to show you some results. This is from the ADNI. I don't know if you guys know the ADNI. It's a public data set of Alzheimer's disease. We took 50 scans from healthy controls and 50 from Alzheimer's disease AD subjects. And they've got 1 millimeter isotropic MP ranges. So if you push them through free surfer, you get like a pretty PL surface and a nice subcortical segmentation. What you can do is to use this as a ground truth and then take the 1 millimeter scan, artificially downsample it, for example, in this case, 7 millimeter coronal. And if you push it through FreeSurfer, like the PL surface looks really terrible, and so does the subcortical segmentation. And then you can train a network to super resolve these guys using our technique. And this is what the output looks like. So it's not identical to the original, but it's pretty good. It's a bit smoother. You lose a bit of like the crispness. But when you push it through FreeSurfer, the results are very, very similar to the original. So to evaluate this in a quantitative fashion, we ran a bunch of analysis for which we kind of like, we know what to expect, right? So we look at the hippocampal volume, which is a very well-known biomarker of Alzheimer's disease. So if you compare the volume of the hippocampi of Alzheimer's and controls using the original 1 millimeter isotropic data, using FreeSurfer as well, you get an effect size of 1.38. And when you start decreasing the resolution, the effect size goes down quite a bit. It falls below 1 at 7 millimeter if you don't do super resolution. But with our technique, you can stay above 1.3, even at 7 millimeter. When you look at the scatter plot, it's very clear what's happening. So this is on the x-axis is the ground truth from the 1 millimeter scans, and the y-axis is from the 7 millimeter scans. And if you don't do super resolution, which is the red dots, first of all, you have more noise. You have a positive bias, which is particularly bad for the subjects with more atrophy. And all these problems are solved with the super resolution technique, which is the blue dots. And that's in segmentation. In registration, we did this tensor-based morphometry, TBM analysis, and analyzing Alzheimer's versus controls. So this is what you get with the 1 millimeter, as you would expect, like faster expansion of the ventricles and atrophy in hippocampal areas, medial temporal lobe, and so forth. When you start going down in resolution, without super resolution, at 3 millimeters, you still have most of the clusters, but at 5, you start like losing a lot of important clusters in the analysis, particularly bad at 7. Whereas with super resolution, like all the clusters are still there. You lose a bit of statistical power, but you get pretty much the same results. This is only for super resolution, right? We also tried the joint super resolution and synthesis using the ADNI. The advantage with ADNI, again, is that you have the 1 millimeter MP regis. So you can use the FreeSurfer segmentation or registration from these scans as ground truth. But you also have this 5 millimeter flare, which is axial, and it looks like this. So it looks terrible in coronal plane, as you would expect. You cannot, FreeSurfer operates on T1s, so you cannot extract surface from this. There is still a module to get a segmentation from known T1 contrasts, and it looks pretty bad because of the lack of resolution. And this is what the jointly super resolved and synthetic T1 looks like. So you recover again segmentations that are much, much nicer and closer to the original. We can repeat the same analysis. This is a harder problem. So your effect size goes down to, more than before, more so, to 0.90, but it's still much better than the 0.26, which is really, really bad that you would get with the flares directly. And this is what the scatter plot looks like. So the flare is the red dots. You have a bunch of outliers, like it's very, very noisy, and super resolution does much better. Same thing for registration. You can do TBM with flare. It's a complete disaster. You still pick up the ventricles, of course, because that's a slam dunk. You could probably pick that up with like 10 millimeter scans, but the rest is very, very noisy. Whereas with this, our technique, you recover pretty much the original maps. And finally, some results from the PACS from Mass General Hospital. We used 40 subjects for this analysis. They have a 6 millimeter flare, axial flare, 6 millimeter axial T2, and a 5 millimeter sagittal T1, as well as a low resolution blurry isotropic scout. So you can actually, because it's got like a SPGR T1 contrast, you can push it through FreeSurfer directly, but it's pretty bad, especially for the cortex that looks like super flat if you pay attention. Also, you can see it here. It's very, very noisy. We also tried a Bayesian method by Mikkel Brutfors and colleagues from UCL, which helps a little bit. It makes things a bit crisper, but it doesn't really solve the problem. And this is what you get with the joint super resolution synthesis technique. So you enhance the contrast quite a lot, and you enable FreeSurfer to do a much better job. We don't have 1 millimeter scans for these guys, so we cannot compare against ground truth, but we looked at correlations with age, and you get exactly what you would expect. So this hippocampal volume going down very cleanly. This is the ventricular volume going up. You can almost tell this quadratic shape that we know exists with aging, this basal ganglia and so forth. And you cannot get these effects with the competing techniques. And just very quickly, I'm going to show you pretty pictures, no quantitative results here. This is super preliminary. Hyperfine is this portable MRI scanner. It's pretty cool. You can just plug it in a normal outlet on the wall. You don't need helium. You don't need like a super strong magnet. It's 65 millitesla, and you get axial images that are not fantastic, but for what it is, it's pretty amazing. I must say, it's just 65 millitesla, which is nothing. They are 1x5 in plane, 5 millimeter thickness. This 32. This is what 31 looks like. And this is what our technique gives you, which I think it's pretty neat if I flip back and forth. Same thing with Epimix. Like I said, this is very preliminary, but I still wanted to show you the pictures. This is a very fast EPI sequence. In 2 minutes, you get less than 2 minutes. You get T1, T2, T2 star, you get flare, and you get basic diffusion at 1x1x3, again, axial. You get the typical EPI distortion here in the frontal lobe. This is the flare scan, the T2, the T1, and this is what you get from our algorithm. I flip back and forth. And just to wrap up very quickly, future work, clearly make this work with pathology. That's just the biggest challenge we're facing at the moment. Like if you're going to run this on images from the PACS, you really need to be able to deal with tumor, stroke, and so forth. Right now, we are not simulating any of this. And of course, right now we're using pretty simple architecture, but there is a lot of like modern CNN architectures that we can try. Like for example, the module that Nalini just presented, also adversaries, progressive CNNs, and so forth. And yeah, I'm gonna acknowledge our funding sources, but most importantly, like co-authors. The best part about science is the people you get to work with. Thank you very much.
Thank you very much, Eugenio, for that great talk. Now I'll introduce our third speaker, Suheyla Karayumak. Suheyla is an instructor at the Psychiatry Neuroimaging Laboratory at Brigham and Women's Hospital and Harvard Medical School. She develops new solutions to diffusion MRI data harmonization problems with the goal of enabling the study of the human brain's microstructure and connectivity in very large data sets. Her other research interests lie in machine learning and neuroimaging techniques to understand the risk factors of developing psychiatric disorders before the onset of the diseases using very large-scale data sets. Thank you very much, Suheyla, for joining us. We look forward to your great talk.
Thanks for the introduction, Lauren. Today, I'm going to talk about our retrospective and prospective diffusion MRI data harmonization strategies, which are critically important for the joint analysis of multi-site neuroimaging studies. Diffusion-weighted magnetic resonance imaging is an non-invasive imaging technique that has been actively used in many neuroimaging studies. Diffusion MRI aims to characterize tissue microstructure and structural connectivity in health and disease using diffusion MRI measures and tractography analysis. Diffusion MRI measures are quite sensitive to the structure of the underlying tissue and have contributed to a wealth of knowledge about the abnormalities in several neurological and psychiatric disorders. Although there are several advantages of the diffusion MRI, the diffusion signal is sensitive to molecular water motion and it can be easily modulated. For example, even the scan time of the day or fluid intake might have some effect on the diffusion signal. When the same subjects were scanned on the exact same scanner twice, we observe around voxel-wise 2 to 4% intra-subject variability in the white matter and up to 9% in the gray matter of fraction anisotropy. On the other hand, when the same set of subjects were scanned with 2 different scanners, we observe large scanner-related differences even with the same sequence used across scanners. We see around 10% difference in FA in white matter, about 20% in gray matter, and up to 35% difference in CSF areas. The measurement differences across scanners can get even more drastically larger when there are differences in vendor, software, or sequence of the scanners, and these can induce nonlinear effects, scanner effects on the diffusion signal. Therefore, scanner-related differences make it impossible to naively pull diffusion MRI data across multiple scanners for joint analysis. The efforts to remove the unwanted scanner-induced effects from the diffusion MRI data are called harmonization. Harmonization can be considered successful. First, after harmonization, data variability is close to rescan or intra-subject variability within the same scanner. Second, if the harmonization preserves the biological variability of each subject during the harmonization process. There are multiple strategies to harmonize multi-site diffusion MRI data. We can categorize the methods into two. First type of methods are data pooling techniques, which aim to statistically remove the scanner differences from the derived diffusion measures. Harmonization is performed in the last step of the pipeline after applying data pre-processing, model fitting, and computing diffusion measures and additional processing steps. It is, however, unclear how design choices for the different data processing pipelines can affect harmonization results. Therefore, we recommend extra caution while using the statistical approaches for harmonization. The second type of approach removes the scanner-specific differences from the diffusion MRI signal, which also includes multiple works of ours. The approaches in this category are applied at the signal level early in the pre-processing pipeline. Therefore, the method allows the use of any diffusion MRI data processing like model fitting or tractography analysis after harmonization. Depending on the availability of the traveling subjects, two primary approaches have been proposed by our group. The first one is the retrospective harmonization. It is for use where data has already been acquired without traveling subject. Therefore, it uses biologically matched controls at the group level across two sites and scanners to learn the scanner differences. And this retrospective data harmonization method is widely used in clinical applications for the retrospectively collected multi-site data sets. On the other hand, prospective harmonization includes state-of-the-art deep learning techniques for harmonization, but it requires traveling subjects scanned at each site to create a within-subject overlap cohort. To capture scanner differences, both retrospective and prospective harmonization methods use rotation-invariant spherical harmonics features. Rich features represent the energy of the signal, the energy of the diffusion MRI signal at different frequencies represented by different spherical harmonics orders. And rich features represent also different microstructural tissue properties which are independent of the orientation. In the figure, you see 5 rich features of the same subject scanned with 2 scanners, scanner 1 and scanner 2. In our strategy, we select one scanner as a reference scanner, and the other scanner to be harmonized or mapped to the reference is selected as a target scanner. We therefore learn the mapping from target to reference scanner using these rich features. Retrospective harmonization includes 2 parts. The learning part is performed using training data that's a subset of at least 16 healthy controls from each site, whose age and sex are distributed similarly across 2 sites. The first aim of the learning is to create the rich feature templates by computing voxel-wise average rich features of each scanner. Second is computing the scale maps of voxel-wise ratio between the reference and target rich features. So in the second part of the harmonization, in the applications step, we apply the scale maps to target diffusion MRI data of healthy controls and patients to appropriately scale the diffusion MRI signal. Our harmonization method also supports different p-values, spatial resolutions across sites, and also supports multi-shell diffusion MRI data harmonization. And our software is available online if you're interested. I also would like to note that given the importance of the multi-site harmonization problem, harmonization community challenges were organized in the past years, and this approach was selected as the best performing method for many diffusion measures in the latest challenge. You can find the results if you're interested in this paper. As I mentioned earlier, the inter-scanner differences can be learned using traveling subjects in relatively quick succession, such that measurement differences can only be attributed to scanner-related differences, hence not to biological differences. This should also allow scanner-specific effects to be captured with fewer subjects. Hence, using traveling subjects, we develop deep learning-based techniques, such as deep convolutional neural network and cycle-generative adversarial network with segmentation loss for prospective diffusion MRI data harmonization. I'm not going to go into details of these approaches, but briefly, these approaches aim to improve the performance of harmonization by learning the nonlinear mapping across two scanners or two sites using the traveling subjects. We have applied the retrospective diffusion MRI data harmonization method in multiple clinical applications. One of these was a large schizophrenia database collected from 13 international studies. This project had two goals. First one was harmonizing the diffusion MRI data, and second one was standardizing the clinical data across multiple sites for joint analysis of diffusion MRI data and clinical data across 13 sites. By leveraging the harmonization, we were able to improve the statistical power in our data, and we also characterized white matter pathologies across the schizophrenia lifespan. Also, we were able to use advanced data science approaches that are suitable for large data sets and identified 3 distinct clusters of fiber tracks. I will not go into the details of the findings, but briefly, using the large sample, we were able to show the dynamic interaction of the pathologies at different ages and stages of the disease along the schizophrenia lifespan in these 3 distinct clusters. We have applied the prospective harmonization to traveling subjects in multiple applications. We first applied this method to 16 subjects, where the subjects were scanned with both 3 Tesla connectome and Prisma scanners. We harmonized diffusion MRI data of Prisma to connectome. On the top figures, you see the fraction anisotropy or FA maps from connectome and Prisma scans. And on the bottom, you see the percentage differences of FA between Prisma and connectome, between 2 scanners, Prisma and connectome, before harmonization and after harmonization, respectively. And even though the voxel-wise percentage differences of FA between these 2 scanners were large and spatially varying before harmonization, these differences were minimized after harmonization. We applied the second experiment on 7 Tesla and 3 Tesla scans, where 100 subjects were scanned with both 3 Tesla and 7 Tesla scanners. We harmonized 3 Tesla to 7 Tesla. You can see the voxel-wise percentage difference of original 3 Tesla and 7 Tesla scans before harmonization for one subject. The voxel-wise differences were almost up to 50% before harmonization, and these differences were minimized and much closer to 0 after harmonization process. So before concluding my talk, I would like to emphasize the important points and challenges that need to be considered by all harmonization algorithms. First of all, harmonization approaches should be validated in various diffusion measures and model-independent metrics. In the figure, you see the performance of our method in the harmonization community challenge for 2 diffusion measures, fractional anisotropy and mean kurtosis. The challenge provided a fair platform to evaluate the performance of the 20 state-of-the-art harmonization techniques. Here, lower absolute relative error indicates better performance, and our method performed the best in multiple diffusion measures. Harmonization methods should also take extra precautions and ensure that fiber tractography is not affected in any way during the harmonization process. In other words, we do not want to introduce new fibers or change fibers' original anatomical orientation during the harmonization process. To demonstrate this, in our earlier work, we ran a tractography experiment to compare the anatomically segmented white matter tracks before and after harmonization, and to observe the harmonization effects on the fibers. We observed a significant overlap between fibers before and after harmonization. It was over 95% overlap, which means that we haven't introduced any new fiber orientations during the harmonization process. What are the challenges of the harmonization? For retrospective harmonization, since we are using different set of controls across 2 sites, nonlinear registration is required them to bring the images to the same space. The performance of registration might be affected when the data has large variability. For example, if registration fails or is not accurate, it can lead to also errors in the harmonization results. This is of particular importance when data has like subjects with large ventricles and subjects with other large variabilities. For prospective harmonization, acquiring traveling subject scans can be usually challenging, expensive, and time-consuming. However, as I mentioned before, an ideal way to learn the scanner differences would be using the traveling subject scans. Therefore, it can be highly beneficial for multi-site studies to understand how many traveling subjects are required. But this is not as easy as it sounds, because it can be highly challenging to standardize the numbers across different vendors, versions of MR software, as well as acquisition parameters. Current harmonization methods can have certain limits on the patient populations, especially if the patients have growing pathology such as tumor or MS lesion. Harmonization doesn't work usually in these scenarios. Future methods must be validated in such data sets too. This is the end of my talk. I would like to thank my mentor, Dr. Yogesh Reddy, my team in PNL, and our collaborators, including Dr. Lauren O'Donnell, as well as our fundings which supported this work. Thanks for listening.
Thank you very much for that great talk, Suheyla. Now I'll introduce our 4th speaker, Yangming Oh. Yangming Oh is assistant professor of radiology at Boston Children's Hospital, where he is a key faculty member of the Fetal Neonatal Neuroimaging and Developmental Science Center. His research interests and expertise are in the domain of medical image analysis algorithm development for registration, segmentation, and pattern recognition, and their applications in translational research. While broadly expert in these areas, he has chosen to focus his efforts on clinically impactful applications of neonatal and pediatric brain imaging. Thanks for joining us today, Yangming.
Thank you, Lauren, for the nice introduction. So in the next 12 minutes or so, I'm going to talk about our very recent efforts on quantifying normal and detecting abnormalities, especially in children and young children, then later across a lifespan. So first of all, I want to draw a clear contrast between what we call as big focal abnormalities, where in the left panel you see 2 columns. Left column, the brain tumor, and right column is the acute stage of the stroke lesions. Those lesions are for us easy for us to visually see it, and you can imagine deep learning and machine learning can detect them relatively easier compared to the right panel. On the right panel, I'm showing some small diffuse lesions. The first row is brain injuries in the first week of life, right after birth. The second row is 4 cases, 4 patients in the first year of life. If I look at the upper left column, upper left corner, people are radiologists are usually looking for really dark spots in this apparent diffusion coefficient ADC maps. Those super dark regions are usually what they call abnormalities. This isn't really the case in this case, but in the remaining 6 cases, if the question is where do you see the super dark regions were abnormalities, especially in those regions pointed out by the arrows, that's more difficult. And if we go back to visit the principle of machine learning, so machine learning learns how the abnormality regions, usually labeled by human experts or multiple experts, how they look like, then come into a patient's image and label every voxel as lesion or normal. You can look at local feature, global feature, or combination deep learning versus traditional machine learning. But those voxels you labeled as abnormal, then you group them after some post-processing, you call them abnormal regions or lesion regions. So that's a principle, but then if we visit how machine learning performs in big focal lesion, left 2 columns, versus a small diffuse lesion, right 2 columns, going to go over it, spend maybe 1 minute to go over it. Left 2 columns, left column, brain tumor, 1 case from the BRATS public data set, and the second column is the acute stage of the stroke lesion, right regions, also from the public data set SPES. The second row, we're looking how big the lesion is in terms, in comparison to the whole brain volume. So the ratio of the lesion, then you can see the average brain tumor patients, their brain tumor lesion occupies around average 5–6% of whole brain, versus in acute stage of the stroke, somewhere between 10 to 15. So roughly above 5 and close to 12–15% of the brain are injured. Those are big focal lesions. And in the third column, I'm showing each dot is reported dice, average dice across all those patients in that data set by 1 study. So each dot is 1 study. And we can gather different deep learning studies and put them into this distribution. But you can see more or less different studies vary by their pre or post processing or by the architecture of the neural network. But roughly, you can see the algorithm can achieve 80–90% of accuracy, dice accuracy compared to the human expert labelings. Those are big focal lesions. On the right 2 columns, small lesions, also labeled as red in public data sets. In terms of how much the brain it has been injured, it's around less than 1 or 2%. Some were very close to 0% of the brain have been injured. So you see this huge imbalance problem. You see a small diffuse lesion where the same thing on the third row, each dot is 1 study, the reported average dice in a deep learning study. Then different studies more or less the same, report somewhere between 20 to 60–65% average dice across all those data sets, across all those patients in the public data sets. So this gray bar, we want to show there is a systematic gap between deep learning segmenting the big focal lesions and deep learning segmenting the small diffuse lesion. Well, there are many things to discuss. So maybe the reference is not right because it's difficult, really difficult for neurologists to label the small diffuse lesion. Maybe the reference is not right, or maybe the metric is not right. Dice is not necessarily the best accuracy metric. And also people are trying to improve in the architecture of neural network. But there are many ways to address this problem. And here I'm showing one way that is where I'm motivated. So one way is to say, if I put my image into a convolutional neural network, I only implicitly characterize each voxel's by looking at the global information in the first couple of layers in the deep neural network. But what if I explicitly put the XYZ anatomic location into the convolutional neural network? The assumption is that different voxels at different locations, even the same intensity, let's say the same intensity or same feature in the left and right brain, because there's a slight difference between the left and right brain, so maybe they should be labeled slightly different, or their probability of being lesioned is slightly different. So here I'm citing two studies, only two studies, focusing on small diffuse lesions, one matter hyperintensity, and they can show there's a slight improvement, roughly around 5 percentile in the DICE improvement if I explicitly add the location information. So what we want to do, that's a background. What we want to do is we want to push this little one step further. So we want to claim that it's not just the location, it's the normal variation at each location. So here I'm going to show X, Y, those two voxels. And in those two voxels, one is labeled green, normal, and one is labeled abnormal, red, because if you look at the normal variation of their intensity at those two locations, left and right brain, they differ slightly. And because one falls within the normal variation, one falls slightly out the normal variation, only very subtle, slightly out, they are labeled different. So it's not just the anatomic location, it's actually one step further, the normal variation in each location. And we have colleagues from UK that just published one brain paper that shows that as well in preterm infants. So all those evidence motivates us, at least this is one way to look at it. We can quantify the box-wise normal variations, then feed that into machine learning. So here is several cases, certain cases from the first week of life. We construct their put their real brains into a construction of the virtual brain, which we'll call the normal atlas. And here's the average intensity, average ADC values. You see slight difference. And the standard deviation at each voxel is also different. Some regions vary more than other regions. Then we can do this not only for the first 2 weeks, we can do that for the first 6 years. So if you link all those things, we very rarely have one person, normal person, coming to us so many times from 0 to 6 years. But if we do this virtual brain, you can call it Adam, you can call it Eve. This is virtual brain shows the average brain across how the average brain grows across time from 0 to 6 years of age. And there's spatial difference, there's temporal difference. Then we can quantify. So for example, different regions of the brain do develop totally different. So if I'm looking at the top of the brain, region 4, and the red curve is how they change, cross-linking all those time points, and the blue curve is the rate of change, which I would say it's less steep than another region, cerebellum in region 7, where you can see the rate of change, the drop is much quicker. And they get to their saturation at slightly different ages, one at 1.5 years and one is post 2 years, a little beyond 2 years. So we can quantify how different regions, at the voxel level, how different regions saturate and when is the saturation age. Then you can see in this figure, green means earlier age and red older age, around past around 2 and 3 years of age when they get to saturate. Then the whole brain actually shows a pattern of growth from the internal of the brain to the peripheral. It's earlier maturation internally, and it's early maturation in the back of the brain than in the frontal brain. So you show this internal to peripheral and back to front growth pattern. And we can separate those voxels to show in the first year, those voxels matured, then next half year, and gradually all those regions get matured. So we do see there's a slight, especially in children's brain. We pick children's brain because they are moving every day, every week, every month. So capturing the normal variation at each voxel level, each regional level becomes more significant. Then coming back to our question, if I have a patient brain, ADC map, I can compare this every voxel to our atlas and convert every patient's ADC map into a so-called Z map, where Z value at every voxel quantifies the standard, how many standard deviations away this patient's ADC value is from the population average. Then this Z map is not only age matched, but also space matched. It carries the normal variation, carries the subtle deviation from normal at every location, anatomic location. So then I can compare. So you can use unit, you can use different architectures, unit plus or any unit or arithmetic. you can use different models. But if I use the same model and compare ADC versus Z versus ADC plus Z, blue is my ground truth by 2 experts. You can see Z has improved significantly over using ADC because we have built into ZMap the spatial and temporal normal variation at every voxel. And if we do the statistics around for across 143 subjects we have, and you can see around 45 percentile dice has a dramatic increase to 75, 30% increase. Then it saturates there, adding ADC. ZMap gives the biggest effect and significant effect. Then we can compare whether this ZMap, this atlas, which we built from 3T, whether it applies to GE 1.5T versus Siemens 3T, and significant difference was not observed, meaning that the atlas and the ZMap and the approach can be applied to different scanners, images from different scanners, at least in our cohort, without any difference. So that's then that motivates us to do more study along this line. So just now we talk about 0 to 6 years of age. We can do lifespan from, we have gathered more than 10,000 MRIs across the lifespan, and we can divide them into 36 finely defined age groups or 10 coarse defined different stages of brain. And here is a slight movie to show we can construct the atlases in every age group. And neuroscientifically, you can see how the brain develops over time. At least the contrast changes, the ventricle grows bigger and bigger as we age. And if you just draw the whole brain volume across different ages, you can see it's almost linear growth in the first 2 years of life. Then gradually we got to 25 years of age where we reached the peak, then gradually reduce and accelerated reduction after 85 years of age. So, but we want to use that for radiology assistance. Then we can test different abnormalities. We also doing efforts to improve neural network architectures and to incorporate multi-channels of atlases. So that's ongoing work. So that finishes my first topic. I want to switch gear for very quickly the second topic. So previously, in the first topic, we talked about quantifying abnormality in every voxel. Now, what if I summarize the whole brain into one metric? It can be the predicted age, it can be the predicted IQ score, it can be other things. So our colleague from UK, James Cole, who is a pioneer in this direction, has a series of publications and a very nice summary. Here I just cite one summary. It's just one of his many publications in this domain. The figure is very beautiful. It shows if I can learn the age information from multiple MRIs, thousands, tens of thousands, I can build a machine learning predictor that can give me predicted brain age. And now you can see if the predicted brain age is older than actual age, it shows accelerated aging, usually associated with some lifestyle changes, smoking, alcohol, or other things, opioid, or maybe there's a disease, or maybe there's an early indicator for some disease. Then in last year, 2 years ago, Nature Neuroscience paper with 20,000 MRIs across lifespan, a group of researchers have shown that different diseases like will drive the brain age, summarized by machine learning, will drive them at different scales. So for example, here's the dementia, here's a mild cognitive impairment, and you can be as subtle as just autism or ADHD. So different diseases are driving the machine learning predicted brain age at different level. So suddenly there are two questions. One, accuracy. You need to be very, very accurate because we're talking about only 0.5 or 0.29 years of difference between the predicted and actual age. So it needs to be very accurate. Another thing is it needs to be very specific. Different diseases or different life environmental conditions would drive the aging of the brain roughly at the same level. How do we differentiate different diseases or different conditions? So sensitivity and specificity are really the key. So along this line, talking about specificity, we want to split the brain into two questions. So some people can show a aged brain, accelerated aging brain, because of morphometry change in large ventricle in this case, predicted age 65. SAMCAS also predict the same age, 65. It shows older, not because of morphometry, but because of contrast change. So there are two channels, morphometry change and contrast change. They can be summarized into roughly the same predicted age or same difference of predicted age from the actual age, but maybe they will give us some signal for the specificity. So along that line, then we design an algorithm that splits the T1 image into two channels, the contrast and morphometry. Then two channels can give us some room to play with attention mechanisms where we optimize or slightly change, adding the attention into neural network. Here, the architecture, I'm not going into the details, but every step of the component algorithm shows that we can reduce the error in the predicted age. Used to be around 4 years of age in various reported studies, but now we can reduce that to 3 years of age because of this algorithm design. So accuracy can increase our sensitivity, and here we're comparing with different just published multi-channel neural network architectures, and we can show we can improve a little bit. So now I want to show our goal in this domain. So here's a table comparing our study with many other studies. And one thing we want to show is big data used to be just 3 or 4 years ago, used to be 1, 2,000, 3,000. And now very soon 10,000, we have gathered 17,000, the very recent over 30,000 across our lifespan. Second thing is across lifespan, because we know the actual age, but a 40-year-old brain can look like 20 or can look like 70. So the predicted age in the brain is embedded in the brain, MRI, is really all across the board. So we need to have a lifespan. We cannot control ourselves to be just 20 to 40 or 40 to 50, because a 40 can look like a 70 brain in MRI. So lifespan, we have almost 0 to 100 years of age, whereas many earlier studies focus on narrower age groups, and very recently we have more and more lifespan studies. So another thing is sensitivity. So if I draw, the bar here shows the age variation, the age range in different studies. Every bar, we are from 0 to close to 100 years of age. And compared to those studies that carry children and adults, we can show the accuracy, the error of our study here. So accuracy is one thing, and then sensitivity. Beyond sensitivity, we want to show the specificity. Different ages caused by different mechanisms, and we want to apply that into different sexes and in different conditions. And we have some data from lifestyle or children born to mother of some diseases. So that's our ongoing work. So I'm going to stop here, and I want to thank my collaborators, my mentors, my group members, and I want to say the journey just got started. And if you have data, you have interest, we're happy to collaborate and welcome you to join us. Thank you. I hand it back to Nora.
Thank you, Yangming, for that very interesting talk. So I'd like to thank all of our presenters, and thank you very much to the audience for joining us at our symposium today. We now have a few minutes available for questions for the presenters. So we're going to do this a bit like a panel discussion. I have some questions that we'll all work together on answering. So our first question is, beyond the individual projects that you described, what do you think is the most exciting future opportunity in neuroimaging for assessing brain health? What are the outstanding challenges in implementing this? And what are the limitations in using machine learning for these projects? So I'm going to call on some of our presenters. If anybody wants to go first, look really excited right now, and then I will pick on you. Okay, I'm going to pick on Eugenio then. What do you think?
Sure. Thanks, Lauren. So, I mean, I think the most exciting future opportunities at the convergence of the availability of bigger data sets combined with the algorithmic advances and also the availability of computing power, right? Like we are now at the point where, like Yangming was saying, like big data is now tens or close to 100,000 scans. Like I said in my talk, we also have like a zillion scans sitting in packs around the world, and we finally have the computing power and also the algorithms to squeeze information out of these images. Now, in terms of limitations, there's a lot of issues with privacy, with sharing data. But at the same time, there is also a lot of work on federated learning, right? To try to train neural networks using these big data sets that combining different institutions without having to transfer data around. So I think we live in a very, very exciting time for machine learning in neuroimaging. But I don't know, what do the others think?
Yeah, let's see. Let's hear from one of the younger investigators with even maybe more of a view of what's the absolute newest cutting edge. What do you think, Nalini?
Yeah, so I think one opportunity that I'm really excited about is, I think now we have a lot of the tools and people are starting to build up the data sets in order to understand how we can specifically design acquisitions to be like as tailored as possible to a particular downstream application. So what I mean by that is that there are a lot of people working on things now, like given you want to image a particular anatomy or a particular subject, can you learn exactly what the sampling pattern should be when you're acquiring a case base? And I think if we do a good job starting sort of that early at the scanner, we'll, as we progress through the pipeline, end up with really high quality results for all of the stages downstream. So like better metrics, better image reconstructions. But then tying into what Eugenio said, I think like the big challenge here is right now, all of these techniques do require a lot of data. And in particular, case-based data or data from the scanner is not often stored. So collecting the data sets in order to do these types of experiments is a big challenge. Or alternatively, we'll need to develop methods which allow us to do these types of experiments, even when we only have access to small amounts of data. And then in terms of the limitations, I think definitely one big thing is we need to be really careful about only applying these methods in situations where we know they work based on how we validated them. So there's like a huge body of machine learning literature on how algorithms trained for specific purposes do poorly when they're applied to populations who aren't well represented in the training set. And I think we all in the MR and neuroimaging community need to be really careful and cognizant of that.
Yeah, those are some very interesting thoughts. So we're thinking about there's more big data, there's better algorithms, and then also we want to have better data starting at the scanner level. So these answers are so far kind of starting to cover the whole spectrum of what is exciting and what the challenges are and what we may improve. Let's see, Yangming or Suheyla, does either of you have something that you'd like to add to this?
I think I agree with the others' points about the big data opportunities and the machine learning applications, and connecting these big data, like clinical behavior cognition data, is exciting. At the same time, now we have daily activity data sets such as Fitbit activity, screen time data sets are available for many studies. But now the big question is, how do we combine all these data sets and relate them to the neuroimaging data? I think it's a big question to answer. Therefore, I think opportunities are very similar to the challenges. How do we analyze these dynamically changing, large-scale, high-dimensional data sets in a more principled way? So deep learning is one way, and it is very promising and offers some solutions, but I think more needs to be done there. At the same time, there are like limitations that we know about. For example, every one of us like talk about these. The deep learning doesn't work on at the moment, like for the gross pathology such as tumor, MS lesion, and more needs to be done there. So yeah, I think these are my thoughts about this.
That's excellent. That's another perspective on it, on the really the rich resources and the rich data that's becoming available. It's very exciting looking forward. Yangming, would you have anything to add to this answer of this question before we move on to another? What do you think?
I'll try. So I think deep learning and big data, for example, 10,000. I just heard yesterday that we are closing to 130,000 MRIs. So that's a way to go. But I want to norm two things. One is, so as I showed in the earlier slides, small and diffuse lesions, deep learning is still not catching up the accuracy as they do in the big focal lesions. So that's one area. But if you want to detect lesions early, you can think of early stage lesions, even tumor has to be small, has to be subtle. So not to wait until a later stage of tumor or lesion. Detect early, I think there are some opportunities there, not just data, but deep learning algorithms. That's one thing. Another thing I was thinking, even big data. So what I just showed in the second one, that's age. Predicting sex. People have shown very high accuracy with 10, 30,000 MRIs. But on the other hand, we also notice in ABCD dataset, 17,000 MRIs. And a bunch of groups across the world were asked to predict the fluid neurocognitive score. And the accuracy was almost lower than random guess. Even with deep learning, various groups reported deep learning results. And I can report their predicted fluid neurocognitive score. The correlation between the predicted score and the actual score is only 0.1, 0.2. And we're talking about 1 being the ideal. You may guess it's close to 0.9, but the actual case is 0.1, 0.2. And there's a bunch of literature there. And also we tried not just the age, but other IQ scores, Bailey scores, or other scores, even with tens of thousands of MRIs. That's another challenge. And some people don't want to share data, so federated learning, where you only share the model or the model of the parameters of the model, you don't necessarily share data. Another thing is, many people are talking about tens of thousands of MRIs, but not everyone has ability to download tens of thousands of MRIs. It usually takes months or years of efforts to harmonize this data, download data, organize the data. So can we have an open cloud central repository where people can access the data, people can search which data set I want. So for example, I just want to correlate gene with MRI. Can I find a subset? Give me 5,000. I just want to look at alcohol addiction, the effect on MRI. Can you give me another 60,000, 6,000?
Sure.
Those ones are the challenges, I think.
That sounds, yeah, there definitely a lot of challenges there. So I broke in a bit because we only have 5 minutes left at the end of our session here. We have several Q&As actually in the written Q&A that people wrote in. We aren't going to be able to address all of them. So if any of you have an answer, take a look at the Q&A and see if there's anything you would like to say specifically. To do one more overall question that is actually also related to one of the Q&A questions. We have a question about how all of these models rely on sophisticated pipelines and networks, and how might they be deployed after training to places outside of the academic medical community that may lack computing resources. And we also have a question from Sung-Ming Hong here that's quite related to that, that says, what do you think we can do to translate these awesome advances to daily clinical practice? A lot of these questions are related to that. Another one said, you know, how can we translate this basically to the population we ultimately want to target? So I think overall, as an overall final question for brief answers in our last couple minutes, do you have any thoughts on translating these to the clinic, to populations in general that we'd like to target, and to places without the computing resources that we may have in the academic medical community? Okay, so let's start with Nalini. Can I pick on you? I see you there first.
Sure. I think this actually in some ways relates to what I was saying in my answer to the last question. At least in my experience, a lot of the really computationally intensive parts of using techniques like deep learning comes in the training, and then in the actual inference time, you don't need so much. So in the setting of limited computational resources, in theory, what we could do is, you know, train somewhere that does have a lot of computational resources and then give models to people to just apply at the site. And then the challenge becomes, like, how do you make sure that the model that you've trained in this other place works for your particular scanner or your particular patient population? Of course, Suheyla talked a lot about what we can do in that regard. I think in general, a challenge for people working on machine learning for these things is either how can we train models which generalize exceptionally well to really, really diverse populations? Or if we can't do that, how can we train models which start at sort of a reasonable initialization so that with just a few examples from a particular site, you can kind of quickly adapt that model in order to be run for whatever target population you're interested in.
I think that really neatly summarizes some of the challenges. Would any of the others have an additional comment on this in our last 2 minutes?
I can
Okay. Oh, go ahead, Suheyla, and then Eugenio next if we have time.
Okay. So I can also suggest using cloud computing when there's like a lack of source for the computing resources. For example, I think cloud computing provides a cheap way of running like high computational tasks in GPU or CPU without any need of buying these supercomputers. And for example, these days, like there are apps that you can upload the data and entire computation happens in the cloud, and you do not need to set up the entire infrastructure in your hospital or your local community practice. So all of these computations can be done in the append, and you just need to pay like hourly prices, and the costs are very minimal. I think that can be done, and all of these setups can be established.
Excellent. Thank you very much. I believe we'll have a closing slide. And Eugenio, if you'd like to mention as we're transitioning to our closing slide, if you have anything to add, please go for it.
No, I was going to say almost the same thing as Suheyla. I mean, the other thing is that if you're using convolutional neural networks for this type of analysis, one of the advantages is that at test time, they are really cheap, computationally speaking, right? Like on the GPU, it will run in like a couple of seconds, but even on the CPU, like on a normal machine, you can get the results in under a minute on a semi-modern desktop. So it's normally not a big problem.
Excellent. Well, thank you so much, all of you, for being here and speaking. And thank you to the organizers of this program. And as you see on this closing slide, the symposium will resume in 15 minutes. So please enjoy this short break. And then you can rejoin using the State of the Field viewing link on your My Agenda registration webpage. Again, thank you very much, everybody, for joining us. This was a very productive symposium session. Thank you. Have a wonderful day.